Hi everyone, I am Dr. Lakshmi, Professor of Microbiology from Sree Balaji Medical College and Hospital, Bharat Institute of Higher Education and Research. Today's video lecture will be on the topic immunity. This comes under the competency MI 1.7. So in today's session, we all will learn about what is mean by immunity, what are the various types in immunity, how do we classify the different types of immunity, and we also will learn about various factors affecting immunity. And we'll learn about few miscellaneous types of immunities like adaptive immunity, herd immunity, local immunity, etc. So these are the specific learning objectives of today's session. Wish you all a happy learning experience. Let's move on to the session now. Immunity. First of all, what is mean by immunity? Immunity refers to the defense power of the host against any infectious organism. So the definition of immunity goes like this. It refers to the general ability of the host to resist a particular infection or a disease caused by the microorganisms and their products. So immunity means the resistance power of the particular organism against any infectious microorganism. So immunity plays a major role in deciding whether the host is susceptible to an infection or the host is resistant to an infection. So this, remember, immunity helps in prevention of the infectious diseases. So how do we classify immunity? So immunity is broadly classified into two different categories. One is innate immunity, that is the inborn immunity. Next is the acquired immunity, which is nothing but the immunity acquired during the lifetime. Innate is further classified into species, racial and individual immunities. Acquired immunity is further classified into active immunity and passive immunity, which is in turn classified into natural and artificial. So we will learn about all these different types of immunities in detail. First is the innate immunity. As I already told you, innate immunity is nothing but the immunity which is present from birth. This refers to any inborn resistance. So this innate immunity is because of the genetic and constitutional makeup of the individual. It does not require any prior exposure. That means even when the organism enters the body for the first time, our innate immunity plays a major role in protecting the host against the organism. It does not require any prior exposure. And it is not modified significantly by repeated exposures to the pathogen over the life of an individual. That means anytime, repeatedly, even if the organism is going to enter, the innate immunity is going to be the same. It is going to give the protection against the entering organism. It is not modified because of repeated exposures. What are the types of innate immunity? Innate immunity is basically classified at three different levels. One is species immunity, second is racial immunity, and third is the individual immunity. So what is meant by species immunity? It is the resistance offered by all the members of a particular species. That means one whole species is either susceptible or resistant to the organism. That means the organism can affect one species but may not affect another different species. This is called species immunity. For example, the Bacillus anthracis bacteria causes anthrax disease. So this bacterium affects the humans, but chickens may not be affected. This is because of species immunity. Next is the racial immunity. So within one species itself, different races may show different susceptibility to infection. So one race may show a susceptibility to an infection, the other race may not show the susceptibility to the same infection among the same species. So this is called racial immunity. For example, high resistance of Algerian sheep to anthrax, and in some part of Africa, some population may be showing a genetic resistance to the Plasmodium falciparum malaria. So this is a racial immunity. Next is the individual immunity. So among individual immunity means different individuals may show difference in immunity towards any organism. Okay, so difference in innate immunity exhibited by different individuals of a particular race is called the individual immunity. For example, homozygous twins may exhibit similar degree of either susceptibility or a similar degree of resistance to lepromatous leprosy or tuberculosis infection. So homozygous twins may show such type of similarity, whereas the heterozygous twins may not show a similar degree of resistance. So this comes under individual immunity. What are the factors affecting innate immunity? So first and foremost important factor is the age. As we all know, extremes of ages are more susceptible to infection. The old age as well as the newborns or the infants, they show higher susceptibility to any infection. Next is the hormones. So hormonal imbalance or hormonal diseases like diabetes or adrenal insufficiency or corticosteroid, if the patient is on corticosteroid therapy, all these things may show more susceptibility towards infection. Next is the nutrition. So there is a complex relationship between malnutrition as well as immunity. So malnourished children are more prone for susceptibility to infection. In malnourished individuals, the humoral as well as cell-mediated immunity may found to be little lesser. So they are more prone for developing any infection. What are the different mechanisms of innate immunity? There are various mechanisms involved behind the immunity. One is the epithelial surfaces, antibacterial surfaces, cellular factors, inflammation, fever, acute phase proteins. All these things may play a major role behind innate immunity. First is the epithelial surface. So epithelial surfaces, they act as a mechanical barrier against any incoming infectious organism. For example, the higher salt content in the sweat and the sebaceous gland secretions and long-chain fatty acids present in the skin, they protect the body against the invading organism. Even in the respiratory tract, the nasal cilia, the cough reflex, all these things will prevent the entry of the foreign organism from reaching them into the lungs and causing any respiratory infection. Like that in conjunctiva, the tears, they play a major role in expelling out the organism which is entering the eye. So like that, the epithelial surfaces will give a good amount of protection. Next is the antibacterial substances. So there are various antibacterial substances present in the blood. For example, the complements, the interferons, the lysozymes, the leucines, plaquins, and many different substances. They not only have antibacterial activity, they also have antiviral activities also. So these substances will protect the host against any incoming microorganisms. They will neutralize the pathogens. Next is a cellular factor. Once the organism crosses these epithelial barriers and the antibacterial substances, the tissue factors or the cellular factors will come into role. For example, the macrophages and the macrophages, they will phagocytose the incoming pathogens, they will engulf them and they will cause intracellular killing of the organisms. Not only these factors, there are some non-specific factors like inflammation which play a vital role in preventing the organism from causing any disease. So in inflammation, there will be vasodilatation, increased vascular permeability which will lead to destruction of the pathogen. Fever is another important factor. There will be rise in temperature which will help to protect the body. And in the blood, in the plasma, there are various proteins called acute phase reactant proteins. These proteins will increase when there is a infectious organism entering the body. For example, the C-reactive protein, the mannose binding protein, all these are acute phase reactant proteins. These proteins will be increased. They will help in protection of the body. They will also help in tissue repair also. So all these different mechanisms play a major role in innate immunity. Next, we'll move on to the acquired immunity. Acquired immunity is nothing but the immunity which is acquired during the lifetime. This refers to the type of specific immunity a host develops after exposure to the suitable antigen or after transfer of antibodies or lymphocytes from the immune donor. So this immunity is acquired during the lifetime. This is not present from birth. So this acquired immunity is basically classified into two different categories. One is active immunity, other is passive immunity. So the mechanism behind acquired immunity is two different mechanisms. One is the humoral immunity and other is the cell-mediated immunity. So humoral response is because of B lymphocytes. They secrete antibodies or nothing but the immunoglobulins. Once the extracellular microbes enter the body, they enter like antigens. So secreted antibodies are released and they will block the infection and eliminate the extracellular microbes. In cell-mediated immunity, the helper T cells and the cytotoxic T lymphocytes play a major role. So the helper T cells will act on the phagocytosed microbes in macrophages. They will activate the macrophage and they will kill the phagocytosed microorganisms. Whereas cytotoxic T lymphocytes, they will kill the infected cells which are having the intracellular microorganism. They will kill the cells as such and eliminate the reservoirs of infection. So in acquired immunity, these B lymphocytes as well as the T lymphocytes play a major role. In acquired immunity, the immune system plays a very important active role in case of active acquired immunity. There are two different types of active immunity. One is the natural active immunity. Because of natural infections, what the immunity is going to be acquired. Next is the artificial active immunity. Because of vaccines, we get the active immunity. So first we'll see about the natural active immunity. So this is because of natural infection. Once the infection enters the human body, so against the infection, our body is going to produce an immune response. So this is called natural active immunity. Remember, this is long lasting and this is the best type of immunity. Among all different immunities, this natural active immunity is the best because this is long lasting and going to give a good amount of protection. Next is artificial active immunity. This is because of vaccination. We simulate the natural infection by giving a vaccine and we are going to induce the artificial active immunity. So vaccines may be of different types. One is live attenuated vaccine, then is killed form of organism, antigens and toxoids. So in live attenuated form, we give the live organism which is being attenuated. So attenuated means you keep on doing a subculture of the live organism so that the organism loses its virulence power. The dangerous power is removed, whereas the antigenicity is retained. So once it enters, it can induce the immune response, but it is not virulent. So this is live attenuated form. Next is a killed form of vaccine where the microorganism is killed by heat, formalin, alcohol and phenol and they are preserved in phenol and merthiolate and alcohol. We also can give toxoids. This is a preparation from toxins released by few organisms. Some infectious disease, the organism as such may not be infectious. They release toxins. Only the toxins are going to cause the disease. For example, in Corynebacterium diphtheria, only the diphtheria toxin is going to cause the disease diphtheria. So in such type of infectious disease, we take the toxin and we deactivate the toxin, so that in toxins the virulence power is removed and their antigenicity is retained. This formulation is called as toxoids. So here we see the various types of vaccines. So first is a live attenuated vaccine. Example, BCG, oral cholera, typhoid vaccines. Among the viral vaccine, we have Sabin, measles, mumps, rubella vaccine, rotavirus vaccines, and 17D vaccines. These are examples of live attenuated vaccines. The major advantage of live attenuated vaccines is here we give the live form of the organism where the virulence is removed. So they will induce a cell-mediated immunity. They can induce local immunity as well as herd immunity, and they are long-lasting. But major disadvantage is because it is a live organism, there is a remote chance of reversion of virulence. And they cannot be administered in all conditions. For example, in immunocompromised people, we cannot give a live organism. And cold preservation is very important. So storage problem is also there. We need to store them properly. So all these things are few disadvantages of live attenuated vaccines. Next is a killed inactivated vaccine. Example, cholera vaccine, pertussis, Salk polio vaccine, influenza vaccine, rabies vaccine. All these are killed inactivated vaccine. The advantage is that there is no reversion because it is a killed form. So there is no reversion of virulence and they are safe in immunodeficient as well as pregnant people. And storage is also not a problem. They are very stable at room temperature. But disadvantage is that they do not induce a good amount of cell-mediated immunity. They can induce only a local immunity. And they are short-lived because it's a killed form. They are short-lived and you need to give more and more booster doses. Next type of vaccine is toxoids, where you prepare the vaccine from toxins. Example is tetanus toxoid, diphtheria toxoid. Advantages, they are highly efficacious and safe. But disadvantages, they are very difficult to develop. Next form of vaccine is subunit vaccine, where the whole organism is not made into a vaccine. Only a portion, antigenic portion, small purified part of a protein portion, which is more antigenic. Only that portion is prepared as a vaccine. This is called subunit vaccine. And example is pneumococcal polysaccharide vaccine, meningococcal polysaccharide vaccines, herpes simplex vaccine, hepatitis B vaccine. All these things are subunit vaccine. They have a low adverse reaction and they are relatively safe. But disadvantages, they are very expensive and difficult to develop. And next is a conjugate vaccine. These are newer forms of vaccine. This is again a type of subunit vaccine only. But sometimes some organism, some antigen may be weaker. So they may not induce a good amount of immune response. So in such a case, we combine a weaker antigen with a stronger antigen as a carrier. This is called as a conjugate vaccine. Example is pneumococcal vaccine 7, pneumococcal vaccine 13 and Hib vaccines, H influenza B vaccine. These conjugate vaccines are more immunogenic. They can induce B cell memory and they can activate Th cells also. But disadvantage is that they cannot induce memory T cells. They are very expensive and relatively difficult to develop. So all these are different vaccines which can induce artificial active immunity. Next we'll move on to the passive immunity. Passive immunity is nothing but in ready-made form we give the immune cells or the immunity. So the definition is the resistance which is transmitted passively to the recipient in a ready-made form is known as passive immunity. Here in passive immunity, the recipient's immune system has no active role. We are going to transfer the immune system, immunity itself in a ready-made form to the recipient. So recipient does not have to do any active immune response. So the major use of passive immunity will be they give immediate and temporary protection in a non-immune host under threat of infection. And in passive immunity is also useful to suppress the active immunity in case of any organ donation and other things. So this is passive immunity. So under passive immunity, we have two different types. One is natural passive immunity, that is from the mother to the infant, the maternal antibodies are transferred. Next is artificial passive immunity, we give like immunization. So natural passive immunity is nothing but this is the resistance transferred from mother to the baby. For example, in human fetus, when the fetus is inside the womb itself, the maternal antibody IgG is transmitted predominantly through the placenta, which is going to give protection to the fetus. Not only that, once the baby is born, the human colostrum through the breast milk, the IgA antibodies reach the neonates and they give protection to the newborn individuals. Next is artificial passive immunity. This is the resistance which is passively transferred to the recipient by administration of immunoglobulins, that is the antibodies or antitoxins or humanized monoclonal antibodies. So all these things can be transferred passively directly to the recipient and you can induce the immunity in the recipient. This is artificial passive immunity. So when you compare active and passive immunity, what are the major difference between active and passive immunity? In active immunity, the immune system is involved. Immune response is present, whereas in passive immunity, no immune response and there is no role of immune system. Antigenic stimulus is required in active immunity, which is not required in passive immunity. So there is a lag phase in active immunity because once the antigen enters, the body has to prepare the antibody. Immune response has to be prepared and exhibited. So there is a lag phase present in active immunity, which is absent in passive immunity. And active immunity effect is long lasting, whereas passive, it is ready-made form, it is a short-lived immunity. And memory is present in active immunity. Once there is a repeated infection also, the same immune response will be immediately presented when there is going to be a second or repeated infection. So the memory is present in active immunity, whereas no memory in passive immunity. Booster effect is present in active immunity, whereas in passive, it is less or no change. Whereas in immunodeficient people, passive immunity is more useful. Active immunity may not be useful in immunocompromised individual. So these are the various differences between active and passive immunity. Let's move on to the miscellaneous types of immunities. First is adaptive immunity. So this is a special type of immunization where instead of whole lymphocytes, only the immunologically competent lymphocytes, they are called as transfer factors. They are very active. So immunologically competent lymphocytes alone are transferred or administered to a immunodeficient individual. This is attempted in the treatment of lepromatous leprosy. This is called adaptive immunity. Only immunologically competent lymphocytes are transferred. So remember, this requires histocompatible donor. Next is local immunity. This is defined as the resistance exhibited against the pathogen at the site of entry. So in the mucosal level itself, this immunity is localized. At the site of entry itself, some secretory IgA, they play a major role. They give a good amount of protection. IgA is present in saliva, mucus and colostrum. They act by two different ways. They prevent the adherence of pathogen. IgA also helps in neutralization of the pathogen. So this is called mucosal immunity. This local immunity, that is the immunity exhibited against the pathogen at the site of entry, near the mucosal area itself. So this is called as mucosal immunity. Last is the herd immunity. This is defined as the overall immunity of the individuals in a community against a particular infectious disease. So this is attained by natural infection or by any ongoing immunizations like pulse polio immunization. So the whole group of people in a community, they are either susceptible or they are going to exhibit a good amount of resistance to a particular infection. So this whole herd immunity may be acquired by the population in the community either by the natural infection or by immunization. This immunity is very useful in preventing epidemics. Herd immunity is otherwise called community immunity. Now we have come to the end of the session. To summarize, in today's lecture, we learnt about the definition of immunity. We also learnt about innate and acquired immunity and various types of these immunities. And we also learnt about the various factors involved in immunity and the mechanisms behind each type of immunity. And we also learnt about miscellaneous types of immunities like herd immunity, local immunity, and adaptive immunity. Thank you all for your patient listening. Hope all of you have understood the various concepts behind immunity. Thank you.